So I ended the previous video with this exact same slide. Again, I want to make sure that you fully understand the difference between all of these. Somatic cells to gametes, diploid versus haploid, autosomes versus sex chromosomes, and homologous chromosomes versus sister chromatids. You should know all of these, and if you don't, please go back and review them before we move on. So, as mentioned in the previous video, we have two parts of meiosis, and they're named meiosis 1 and meiosis 2. And so they have very clear roles within the overall meiosis process. So meiosis 1 is all about a separation of our homologous chromosomes. So what that means is we are reducing our cells from a diploid stage to a haploid stage. All right, that's a huge step because our sex chromosome, our sex cells have to be haploid. Now, meiosis 2 is a separation of sister chromatids. Now you're understanding why it's important for you to understand the difference between our vocabulary here. And so our haploid cells then divide into only having an individual chromosome. So in other words, meiosis 1, we start out with two X's, and those X's are separated. In meiosis 2, that X starts out together, but then ends up being separated, right? So if you think about this, two X's, right, are just made of four different lines. That's what we're kind of ending up with in meiosis 2. So as we move forward, you're going to be able to see a lot of comparisons between mitosis and meiosis. And again, I'll go over that in the very end. So don't get too hung up on that. So like mitosis, we have interphase. The cells have to grow and make proteins. Remember, it's separated into three different parts, right? Our G1, where it's growing and making proteins. S is the chromosome replication, and G2, kind of continuation of G1. This is very similar to mitosis. There's very no differences. So at the end of interphase, you have two full sets of duplicated chromosomes. So it is diploid at this stage. Now, when we enter prophase 1, again, a lot of the same things end up occurring. So the nuclear membrane breaks down, the centrioles make spindle fibers, you can see our homologous chromosomes, you can see our X's to the right. Now, a new word's going to come in, a tetrad. So a tetrad is what we're seeing to the right. So we see four X's, four different pairs of sister chromatids, or two pairs of homologous chromosomes. Now, crossing over. This is something that is huge that we're going to talk a lot about. This is a very simple concept, but so important. So crossing over, it can happen between homologous chromosomes. This is where different genes can get swapped. So there's an interaction between the chromosomes where ultimately information can get swapped. And we're going to talk a lot about this a lot more in the next slide. So during prophase one, the chromosomes are kind of all lined up and kind of right next to each other. So each pair is kind of, holding hands in a sense. Um, so these chromosomes can kind of get tangled up. So as you can see to the right, originally the red one was all red and the blue one was all blue. So in crossing over, they can kind of give each other appendages or give each other chunks of DNA, and kind of mix things up a little bit. And so this is a permanent swap. Once the swap happens, the DNA goes from one to the other. That stays that way. Um, this is how we get new combinations of genes. This is the reason why I'm saying it's so important, because this crossing over has led to the vast majority of our ability to evolve, going from single cellular organisms in the water to humans. It's all about crossing over. All right, so getting back to our different stages. So again, we have metaphase 1. And again, be careful because the names are going to be very similar. So take note of whether it's 1 or 2. So this is metaphase 1. The homologous chromosomes line up in the middle of the cell. Again, in pairs, as you can see to the right. See that the spindle fibers are still attached to the centromere. Remember, that's the center of the X. The centrioles are, again, what produce the spindle fibers. That should all be a review for you. And we have anaphase 1. So our homologous chromosomes separate. Okay? So the individual X's aren't separating. The pair of X's are separating, getting pulled to opposite sides of the cell. But as you can see, the sister chromatids, so the two parts of the X's stay together. Now, we have telophase and cytokinesis all in one slide, so there's a lot of information here. So similar to last time, you have two nuclear membranes forming on opposite sides of the cell. The cytoplasm dividing, that's where the cytokinesis comes from. That's where we have the cells ultimately, if it's an animal cell, it's I guess it would be an animal cell because that's sexual reproduction. It's pinching together in the middle and leading to two cells. So as a result, we have two haploid daughter cells with duplicated chromosomes. Okay? But they are different sets from the original. Okay, so these are no longer the same as the original. So they are different pairings than the original cell. That's really important to remember. So they are no longer identical, especially because they're now haploid and not diploid. All right, well, that was the end of the first part, meiosis 1. Now we are starting meiosis 2. So again, take note of the top. It is now prophase 2. So you're going to see this kind of a repeated process. So the spindle fibers attach to the centromeres of our sister chromatids, so our X's. And the nuclear membrane breaks down. Again, that's nothing new. That's what we've already experienced so far in all of our prophases. Right? And you should be saying to yourself, well, in metaphase, that's where the chromosomes line up in the middle. And that's exactly what's happening. Our X's, our sister chromatids, are lining up in the middle, just like all of our other metaphases. However, this is where our X's get pulled apart. This is where our sister chromatids now become individual chromatids. So our chromatids are pulled apart and then pulled to the opposite ends of the cell. So, telophase 2 in cytokinesis. So our nuclear membranes form again, spindle fiber dissolves, and our cytoplasm divides. All the same stuff. But here's the biggest difference. We end up with 4 completely unique haploid daughter cells with 1 chromosome each. All right? Each one of these cells is completely genetically unique. Okay, that's the whole big point here of meiosis. We end up with unique cells. That's why if you're a girl and you have a sister, the two of you are very different, right? A big part of this is because your mom, each one of her egg cells is different. Obviously, a big part of it is your dad's genetic information, but your mom, each one of her egg cells is very different. All right. As you saw, there are a lot of similarities. So I'm going to quick go through a comparison between mitosis and meiosis. I want your goal should be able to really separate the two of these processes. So, what they are. Mitosis is the creation of diploid somatic or body cells. It happens throughout your life, all throughout your body. It's all about growth and repair, right? You're growing or repairing dead cells or cells that you've lost. It's all about prophase, metaphase, anaphase, telophase, but only once. We only go through that process once. And the whole result are 2 identical diploid somatic cells. They're the exact same. You would never be able to tell the difference between the 2. And this is considered asexual reproduction. Now, that was mitosis. That was what we went over last set of videos. Now for meiosis. You should be able to go through this and know this before I say this. So if mitosis is diploid, meiosis would be haploid. So haploid sex cells. Okay? When it happens, so this is a unique thing, and we didn't go over this yet. For females, it happens before you're born. So when you're still in the womb, all of your egg cells are being created. So you have no new egg cells being created throughout your life. You're actually losing them throughout your life and getting fewer and fewer. Now, males, on the other hand, are very different. Sperm cells are being created all throughout your life. Every day there are more and more sperm cells being created. So very different processes here. So where, instead of being all throughout our body, in women, it's only in the ovaries, and men only in the testes. So the why, it's all about making babies. It's all about producing offspring. That's the whole purpose, and you're making offspring that are unique and different. Now again, mitosis goes through our PMAT, prophase, metaphase, anaphase, telophase, once, whereas meiosis goes through it twice. And you end up with 4 genetically unique haploid gametes. So again, very, very different. 2 additional cells that are unique, and they're haploid. And again, this is called sexual reproduction. So you should be able to get this and feel comfortable about it, and understand this. And so we're going to talk a lot more about this as we move forward with class.